Hi guys, I'm Kunal, a member of the Neuroplasticity team. Today, I wanted to talk to you guys about the awesome neuroscience research going on here at Duke University related to movement in flies. We sat down with Dr. Nina Sherwood to talk about her research in this field, the spastin gene, HSP, as well as the rest of the really cool work her team does here at Duke University. Dr. Sherwood is a professor of biology at Duke University. Her lab studies microtubules.
Specifically, we're looking at the proteins that make up the cytoskeleton. So one of these proteins is called microtubules.
The cytoskeleton, or cellular skeleton, is the network of proteins inside a neuron that gives it its structure. Microtubules are one of the three types of proteins that make up the cytoskeleton. They are made of smaller structures called tubulin that form very sturdy cylindrical structures like this. But that's a little too hard to draw, so we'll just stick to showing them as these green lines. Microtubules also serve as the tracks along which proteins and other cellular structures are shipped back and forth within the cell. How a cell grows, develops, and functions is all critically dependent on these microtubules. Dr. Sherwood's lab looks at the genes related to microtubules and to other proteins involved with making sure microtubules function properly.
One of the genes that we study in the lab is called spastin. And spastin is a protein that we know is important for regulating these microtubules.
One of the reasons that we know this protein, spastin, is important is that when it's mutated in humans, these humans develop a disease that causes people to have difficulty walking. This disease is known as hereditary spastic paraplegia, or HSP. So perhaps a poor regulation of microtubules, which again is the job of spastin, is what is causing HSP in humans. Dr. Sherwood helped to discover that flies also make the spastin protein, and she found that when flies have mutated spastin, they have difficulty walking, just like humans with HSP.
And so what we wanted to do was to take this one step further and to show that human spastin and fly spastin are probably doing the same thing, even though, again, one is a human protein and one is a fly protein.
So now, to see if the fly and human spastin work the same way in cells, Dr. Sherwood decided to replace mutated spastin in the flies with a normal human copy. Hopefully, the human copy would allow flies to walk normally. Sure enough, the flies recovered their motor function. To show even more proof for the similarity between human and fly spastin, Dr. Sherwood tried another experiment.
And then the next thing that we did was to take mutant forms of this human spastin. So these are mutant forms that are based on mutations that have been identified in humans with this disease. Okay, and then put it back in the flies and to ask, okay, now do we give these flies the disease, i.e., do they now walk poorly again?
So, would replacing a normal fly spastin gene with a mutated copy from a human who actually had the disease cause the fly to have HSP symptoms?
And sure enough, we saw that that was also the case. And if we put in a mutation that has worse consequences in humans into the fly, then we would get worse consequences in the fly.
By demonstrating how functionally similar fly and human spastin are, Dr. Sherwood revealed that flies could be used as a model for humans when studying the relationship between spastin and HSP. Lastly, Dr. Sherwood's lab also did an experiment called a genetic screen.
This enabled us to identify, well, really two genes that we could never have just guessed at, right? So by doing this sort of blind search and just asking what is going to help this problem that we see in these spastin mutant flies, we identified these two new genes.
Dr. Sherwood discovered that mutating these two genes would actually make spastin mutant neurons healthier. Surprisingly, these two genes were found in glia, a different cell type in the nervous system that wasn't thought to be involved in spastin function or in causing HSP symptoms.
So I'm really excited about this because it just throws a whole new light on the disease and a whole new way that we can target it.
So in the end, Dr. Sherwood's original research question wasn't about curing a disease or even coming up with a new kind of treatment. It was about understanding one of the fundamental processes that happens in cells, the regulation of microtubules by a protein called spastin. But as we've seen, studying this very specific process may potentially lead to a new understanding of the disease, hereditary spastic paraplegia. By continuing to study neurons in flies, Dr. Sherwood's team hopes to further improve our understanding of both cellular mechanisms and associated diseases. For more information on Dr. Sherwood's research, visit her website, sites.duke.edu/Nina Sherwood Lab.